And now at this time, I would like to introduce and turn it over to Dr. Marcel Mesulam. He is the director of the Mesulam Center for Cognitive Neurology and Alzheimer's Disease, Chief of Behavioral Neurology in the Department of Neurology, Ruth Dunbar Davey Professor of Neuroscience, and Professor of Behavioral Neurology in Weinberg College of Arts and Sciences. Dr. Mesulam, thank you for joining us.
Thank you, Lisa, and congratulations for this wonderful organization. How many people are registered, Lisa?
We were up to 492 when I last checked.
Okay, so greetings to all 492 of you. For the past 26 years, I have been looking forward to the morning of our annual Alzheimer Day event when I would start to walk from my office to the Feinberg Pavilion and get on the elevator to go up to the auditorium where the Alzheimer Day events would be held. And on my way up, I would cross dozens of individuals waiting in line to register for the event. I would walk into the auditorium and have the pleasure of greeting friends and colleagues, meeting new people who hadn't been to the Alzheimer Day event before. And then I would walk towards the podium to start my introductory events. I would turn around and see this beautiful, huge auditorium that contained more than 500 seats that would slowly start to fill up. And then I would feel tremendous pride at this remarkable coalition that Dr. Darby Morhart and her colleagues had been able to put together. So in a few minutes, this auditorium would have hundreds of people standing there shoulder to shoulder, representing our patients, their families, community members, clinicians, researchers, administrators, advisory board members, and they would all be participating in this event. For obvious reasons, we're not able to reproduce this experience this year, but we've done the next best thing, and to hold the Alzheimer Day, the 27th Alzheimer Day, virtually. And looking at the size and the composition of the audience, I have every reason to be as proud today as I've been in the past, but it's not the same thing. It's just not possible to virtually reproduce that electricity of shared engagement in a large auditorium, or to reproduce the spontaneity, excitement of the question and answer period. So as you can imagine, I can't wait to meet and greet each one of you individually on the 28th Annual Alzheimer Day, which is already set to happen on the 5th of May, 2022. 14 months ago, I sent an email to the more than 60 members of our center and asked them to go home and stay there. So you may be wondering what in the world we have been up to in these past 14 months at home. And the answer is different for each individual. Some were homebound because they had kids who couldn't go to school. Some others were declared essential personnel and were allowed to come into the laboratories to continue experiments that couldn't be interrupted. Still others faced unreliable or unavailable public transportation. And over time, even though we're by no means out of the woods, things did start to thaw. And I must say, and I know this sounds strange, thanks to the dedication and creativity of every member of the center, we have come out of these 14 months with flying colors. Patient care was never interrupted, and we now have a hybrid system. Some patients, an increasing number of patients, are being seen in person, and some others to telehealth. Our inpatient research is starting to pick up. Laboratory research under the leadership of Drs. Cengiz Geula and Margaret Flanagan has basically continued without missing a beat. And the interruption of our routine must have encouraged many of us to put our thoughts on paper. And again, I must say that we've been more productive in publications in the past years than we've been in a long time. And there are two papers that I want to mention. In one of them, we found that patients with primary progressive aphasia are resistant to the memory impairment caused by Alzheimer's disease. And in another research project led by Dr. Tamar Geffen, we were able to show that there's a small number of individuals above the age of 80 who are not only resistant to age-related changes in memory, but also are resistant to the emergence of the wear and tear that occurs in memory centers of the brain with time. So these two studies show that memory loss is not inevitable either in Alzheimer's disease or in the course of aging. And we're obviously looking for factors that make that possible. We've also been very productive in designing new innovative experiments and applying for funding. Within the past year, we will have applied for more than $50 million of funding with the NIH. As you know, I established the federally funded Alzheimer's Disease Center here at Northwestern in 1996. And those of you who are regular attendees at our Alzheimer Day events may remember my periodic complaints every 5 years about the rigors of competitive renewal, where our very stern peers looked at what we had done in the past 5 years and decided whether we'd done a good job and deserved to be refunded or not. This year was also competitive renewal year, and I'm glad to tell you that this proposal for renewal under the leadership of Drs. Robert Vassar and Sandra Weintraub is almost certainly going to be successful. So our Alzheimer's Disease Center will be at least 30 years old. We have also been active in interventions and therapy. Our iconic buddy program, as I understand it, has even grown during the pandemic with the senior and junior buddies meeting and working virtually. One of our faculty members, Dr. Borna Banakdarpour, has been developing music as a potential vehicle for treatment in more advanced cases of dementia. Clinical trials under the leadership of Dr. Ian Grant has continued to be active, including in the A4 trial, where the goal is to prevent the onset of Alzheimer's disease. There's also a lot of activity in the wide world of therapeutics. There are very encouraging signs that gene therapy may be effective in patients with FTD caused by C9orf72 mutations. And in the area of Alzheimer's disease, as you know, for the past 20 years, there have been many attempts at decreasing amyloid in the brain that didn't succeed, but there may be an exception. There is a substance known as aducanumab that if you haven't heard about, you will hear about. And aducanumab went under 2 clinical trials. The FDA requires clinical trials to show efficacy before approving a medication. In one of these trials, aducanumab definitely failed. In the other one, it seemed headed for failure, but then reanalysis of the results suggested that there may be some efficacy, but it was small, only 23% improvement in clinical function. Now, you may say 23% is better than nothing, but the problem is that this is an onerous treatment. It requires monthly infusions in the hospital and has potential side effects that require periodic MRI scans, and it is likely to be exceedingly expensive and not reimbursed by Medicare or private insurance. So many of us in the field are on the fence, and I must say, if I were on the FDA panel, which will be meeting about this next month, I'm not exactly sure which way I would be voting. As you know, 3 years ago, we moved into sumptuous 12,000 square feet of space made possible by the remarkable generosity of the Davy Foundation under the leadership of its executive administrator, Mr. Craig Grannon. This allowed every component of our center to come together and sit next to each other so we could learn from each other, teach each other. But the clinic didn't keep up pace with this. It was small and somewhat drab. As we speak, there is a brand new clinic that is being built for us. This will double our space and greatly improve our ability to serve our patients. So, as I hope I've been able to show you, the state of the center is really good. If this had been a usual annual Alzheimer Day, this is the time when I would have been inviting you to join me for lunch during the poster session. This year, all I can do is to give you a rain check, and we will do our best to make up for it next year. A very important part of our annual Alzheimer Day is the presentation of the Duncan Award. So I'm going to invite now Dr. John Disterhoft, who is the head of our pilot projects grant, to present this year's Duncan Award. John.
Thank you, Marcel. It's a real pleasure to present the Duncan Award for the best AD poster, which we choose each year. Our awardee this year is Irfan Tafai, who happens to work in the Mesulam Center. Irfan, as well as his colleagues, Ivan Ayala, Tamar Geffen, Margaret Flanagan, Emily Rogowsky, Marcel Mesulam, and Shangiz Gayula, have done a study entitled Cultured Microglia from Cognitive Superagers Show High Rates of Proliferation. As we're going to hear much more about during the Mendelsohn Lecture from Dr. Roglaski, the Superaging Project was established to identify factors that contribute to the superaging phenotype. These are aging individuals 80 and above who have memories much better than their cognitively normal peers. And the purpose of this particular project was to look at the possibility of a central role for microglial function in the pathobiology of cognitive decline. And they sought to determine if microglia cultured from brains of superagers displayed different characteristics when they were compared to those of normal controls. And I might mention that microglia are supporting cells in the brain. They help to support neurons in the brain. So microglia cultures were made from prefrontal cortex of superagers and normal controls. And what Irfan and his colleagues discovered was that there was a significant enhancement of the proliferative capacity of microglia in superagers. And this may well relate to the protective function of these cells in the brain. This is an important insight into another insight into what makes the brains of superagers different from those of their normal peers. So I want to congratulate Erfan and his colleagues for this really impressive poster. You'll be able to view it as part of the program here. And congratulations again. And now I'll turn the podium back to Dr. Mesulam, who will introduce our Mendelsohn lecturer, Dr. Rogowsky.
Thank you, John. And I also want to remind everyone that the Duncan Prize tradition at Alzheimer Day was introduced by our dear and late colleague, Arye Rutenberg. And this is a tradition that we have continued for many years. So we now come to the crowning glory of the Alzheimer Day program. I want to start by thanking Linda and Bob Mendelsohn for their tremendous generosity and friendship over the years. I should say over the past 27 years, ever since this program was first started. And the Mendelsohn Lecture was established by the Mendelsohn children to honor their parents' 50th wedding anniversary. Over the years, it has become a prestigious lecture that has attracted luminaries in this field, and today's lecturer is no exception. It's really a unique pleasure for me to be introducing Dr. Emily Rogowsky as this year's Mendelsohn Lecturer. Dr. Rogowsky is professor of psychiatry and the associate director of the Mesulam Center. Emily single-handedly put Northwestern University on the map when it comes to research on unusually successful cognitive aging. If Alzheimer's disease can be considered as an example of unhealthy aging, the group of superagers that Emily has recruited can be considered as exemplifying what ideal cognitive aging should be like. Emily has many other accomplishments. She has been a leader and driving force behind our primary progressive aphasia programs, especially when it comes to its imaging and also the atypical type of Alzheimer's disease that causes primary progressive aphasia. In medical schools, it has been traditional to pay lip service to the importance of translational medicine. Emily is an example of what translational medicine should be like. As we started to see more and more primary progressive aphasia patients who came to us from around the country, from over 30 different states, it became clear that when patients went back home, they were unable to access expert speech therapy facilities. So Emily came up with this innovative idea of giving speech therapy online to patients in their home. And she didn't stop there either. She could have just done that and that then gathered anecdotal evidence that the patients just simply liked it. But she turned this program, which she called Communication Bridge, into a rigorous research project that received funding from the Association for Frontotemporal Dementias and from the National Institutes of Health. This is really a model program for translational medicine. I'm not a great fan of Zoom, but this is one exception. This is a great idea to serve our patients who live throughout the United States and in other countries as well. As you know, one of the great revolutions in the field of aging and dementia research is the advent of biomarkers. which now allow us to make an accurate diagnosis in living patients rather than waiting for an autopsy. And two very important modalities of biomarkers are tau PET scans and amyloid PET scans. And Emily pioneered the use of these molecular imaging biomarkers here at Northwestern University. She single-handedly introduced these modalities of molecular imaging to the medical school. Her work is recognized nationally and internationally. She is a frequent guest at summit meetings of the National Institutes of Health. And she is also on the executive committee of the International Society for Frontotemporal Dementias. So I hope I've been able to convey to you the admiration that I have for Emily's work. And now I invite you to do whatever a webinar allows you to do to join me in welcoming Emily as this year's Mendelsohn Lecturer.
Wow, thank you, Dr. Mesulam, for that kind introduction and for the invitation to give the Mendelsohn Lecture. It's truly an honor. My name is Emily Rogowsky, and I'm delighted to welcome you to the 27th Annual Alzheimer's Day at the Mesulam Center. I wanted to start and also end my talk with the word gratitude. We are grateful for the funding that enables our research, including building a passionate research team. Listed here are the colleagues who directly contributed to the data that I'll share today, but there are many other key partners in this work. We are deeply dedicated to the why and the how of aging and dementia research so that we can identify personalized care options that address the symptoms as well as the underlying disease for those living with dementia and their families. I want to give special recognition to our research participants for their time and commitment to research. Without them, none of this work would be possible. For the rest of our time together, I'll share about the what and the how, but also the why behind our work. We are part of a larger network of centers funded by the National Institute on Aging. The green states on this map up here on the right show the geographical distribution of actively funded Alzheimer's disease research centers, and the blue states show the newer pilot centers joining the fold. This network of centers is designed to connect with individuals in their local communities and invite them to participate in research across the spectrum from healthy aging to dementia. Through our research, we collect uniform information so that the sum is greater than the parts. We've enrolled and followed over 2,000 individuals at our center and more than 40,000 participants across all centers. We collect uniform information across sites, which enables us to translate this information into improved understanding and ultimately care for those living with Alzheimer's or related dementia. One of my favorite things about the Alzheimer's Disease Center Network is that each center gets to have its own personality, flair, or area of specialty. The theme of heterogeneity and aging and dementia has been our focus from the beginning, now for more than 27 years. It is a theme that is increasingly being appreciated given the complexity of Alzheimer's disease and related dementias. While neurobiologically, these neurodegenerative diseases may be defined by the misfolding of proteins, we appreciate that the lived reality and experience is much different. These are diseases that are neurobiologically complex. They disrupt families and family relationships. They steal memories and they alter life plans. Our center is uniquely organized with a multidisciplinary structure to tackle these challenges, with researchers and spanning multiple disciplines and specialized research programs examining the cognitive profiles in aging and dementia, the neurobiology of disease, as well as programs focused on optimizing quality of life. We have a clinic and clinical trials and life enrichment programs designed to meet the needs of families. Our diversity of expertise and multi-pronged approach allow us to simultaneously examine these terrible diseases from multiple lenses so that we can find solutions that are practical and meaningful from both a biologic perspective as well as one's lived experience. In these next slides, I'll unpack some of the history and heterogeneity and complexity we have identified in dementia and how this knowledge is essential for identifying treatments and care plans. nomenclature, or how we name Alzheimer's and related dementias has long been a source of confusion. Let's take a look at a simplified history of the shifts in how we've thought about aging and its relationship to dementia over time. While Dr. Alzheimer's first described Alzheimer's disease in the early 1900s, the prevailing view at that time was that senility was a normal and inevitable part of aging. It wasn't until the 1970s when dementia was recognized as a disease and distinct from that of normal aging. The term dementia is defined as a general term for the loss of memory or other thinking skills that is so severe that it interferes with daily life activities. We now appreciate that there are many dementia syndromes, each associated with a distinct clinical initial profile. I've listed 4 of these dementia syndromes on the right here under the umbrella. Alzheimer's dementia is associated with primary impairments in memory, primary progressive aphasia, originally described by Dr. Mesulam in the early 1980s, is associated with loss of language, but relative sparing of other cognitive abilities over time. And then posterior cortical atrophy is associated with visuospatial impairments. Behavioral variant frontotemporal dementia is associated with changes in personality and judgment. Our center is a specialty site for these less common dementia syndromes. Later today, we're going to hear from 3 families, one living with a diagnosis of early onset Alzheimer's, one family living with PPA, and a 3rd family living with behavioral variant frontotemporal dementia. Our center has played a critical role in defining these dementia syndromes and in recognizing their unique care needs associated with each type of dementia. So let's walk through what we've learned and about some of their similarities and differences. When we look under the hood, so to speak, we see that there are differences beyond the clinical phenotype or primary symptoms. And these differences have important implications for developing treatment, understanding the neurobiology, as well as considerations for care management and family dynamics. Let's compare Alzheimer's dementia to primary progressive aphasia. We've already reviewed these first 2 points, but we also see that the risk of developing Alzheimer's disease is greater in women, and this is still poorly understood, greater in women than men, but similar between the sexes for primary progressive aphasia. The age of onset tends to be under the age of 65 for PPA and over the age of 65 for those with Alzheimer's dementia, which means these syndromes are potentially occurring at different stages of life. The majority of cases are sporadic for both syndromes. Insight into symptoms tends to be intact for PPA, but can be compromised for those with Alzheimer's dementia. We have shown that the risk factor profile for PPA is different from that of Alzheimer's dementia. The 2 biggest risk factors for Alzheimer's dementia are increasing age and having 1 or more E4 alleles of the ApoE gene. You see, everyone has a pair of alleles in the ApoE gene, 1 from mom and 1 from dad, and they come in 3 varieties, 2, 3, and 4. If you have at least 1 copy of the 4, it increases your risk. For simplicity here, I'm showing the percentage of individuals who have at least 1 copy in Alzheimer's dementia. We asked the question, is this risk also the same in primary progressive aphasia? And the answer is no. We found that the percentage of individuals with PPA with an E4 was similar to that of healthy older adults. These data suggest that the genetic risk for PPA is different from that of Alzheimer's dementia. We also showed that individuals with PPA and their 1st-degree family members are more likely to have a history of learning disabilities. This observation provides clues about what might make the language network vulnerable to disease. So if neurodegenerative disease is looking for a place to take up residence in the brain, it may target more vulnerable or weaker locations. Family history or a personal history of learning disabilities may provide such a vulnerability. Collectively, these data tell us there cannot be one simple risk score for all dementias. Instead, a more personalized approach is needed. What is causing these changes in memory and language and other thinking skills and abilities, and how do we measure these changes? So in an ideal world, there would be a 1-to-1 relationship with the clinical syndrome and the underlying diseases like this. There would be a PPA disease that caused primary progressive aphasia and an Alzheimer's disease that always caused Alzheimer's dementia. Unfortunately, the reality is quite different. Instead, there are probabilistic relationships between the clinical syndrome on the right and the neurodegenerative diseases shown on the left. This is not a misdiagnosis. And the diseases on the left target, the diseases on the left target different networks of the brain and cause these symptoms. In fact, this slide shows a simpler reality than the number of different neuropathologies and subtypes of neuropathologies that actually exist. This reality has important implications for treatment. Historically, identifying the linkage between these two sides of the slide has been difficult because the right side is based on symptoms during life, and the left side of the screen is confirmed after death by looking under the microscope. The inability to identify underlying pathology during life has negatively impacted early clinical trials of Alzheimer's disease in the past. For example, if a research team developed a purple pill to target Alzheimer's disease pathology, there was no test to ensure that only Alzheimer's disease patients would be admitted into the trial. Looking back now, we know that these early trials included individuals with other neurodegenerative diseases. The goal is to move forward to a situation like this, where the purple medication is only given to the individuals with Alzheimer's pathology. Over the past decade, this dream is now becoming a reality in Alzheimer's disease, where there are emerging biomarkers mentioned by Dr. Mesulam that are emerging to detect the underlying pathology during life. Here is a brief overview of 4 types of emerging biomarkers for detecting Alzheimer's disease during life. Alzheimer's disease is defined by the accumulation of 2 abnormal proteins, amyloid and tau. Amyloid PET and tau PET, shown on the left side of the screen, are specialized brain scans that are able to detect the amount and the location of these abnormal proteins. CSF tests obtained from lumbar punctures and blood tests are listed on the right, are now providing proxy measures of abnormal amyloid and tau protein, but don't show the location in the brain. Blood biomarkers are the newest to the field and are going to be much more cost-effective and less burdensome than other biomarkers. We're really excited about their development. Advances in the identification of biomarkers for Alzheimer's will enable earlier detection of the disease and accelerate the development of new treatments by enabling clinical trials to specifically recruit individuals with the brain changes experimental therapies target. This challenge that I mentioned just a little bit earlier with the pills. We also use MRI scans of the brain to track changes in neurodegeneration or brain health. In my lab, we use a specialized program called FreeSurfer that helps to label the outer surface of the brain, shown in red here, and the gray-white junction, shown in yellow. This thickness between the red and yellow lines provides a proxy measure of the health of the brain. The outer layer of the cortex is like the bark on a tree, and it holds our brain cells. When we compare the thickness between individuals and groups, we can gain precise estimates of how much change is occurring, where it is occurring, and where it is occurring across the brain, as shown in this image here, where the red and yellow colors represent where thinning is occurring across the brain. We can visualize where changes are taking place and quantify this change initially and over time. In PPA, we had observed that the rate of clinical decline was variable from person to person. We then asked the question, does the type of pathology play a role in rate of decline? In this study, we used amyloid PET biomarkers to identify 2 groups, one with PPA due to Alzheimer's and the other due with PPA not due to Alzheimer's. For those with PPA due to Alzheimer's, we found that atrophy was more widespread early on, as shown in the purple and blue, and that the pace of decline was more rapid, as shown in the red and yellow, for those with PPA-AD versus the non-Alzheimer's forms of primary progressive aphasia. These changes were also linked to greater impairments in activities of daily living. So the type of neuropathology seems to be one important factor in determining the rate of disease progression and clinical symptoms. We also wanted to better understand the order in which the abnormalities occur in the brain and their relationship to clinical symptoms. Imaging biomarkers are useful for providing a window into the location and the order of brain changes, which can be linked to changes in cognition. Previous studies in Alzheimer's and dementia, Alzheimer's dementia due to AD suggest Increases in abnormal tau have a close relationship with atrophy and also with changes in memory. We wanted to know if these same relationships hold true for PPA caused by Alzheimer's disease. Adam Mardersteck, a former graduate student in my lab and now postdoc at Berkeley, led this study and found that the answer is yes. Atrophy shown in red and yellow has a similar distribution to tau accumulation shown in blue and green, with the most prominent changes occurring in the left hemisphere, which is known to be important for language. The bottom image here shows that changes in atrophy and tau PET highlighted in red, orange, and yellow are associated with decline in naming ability, a primary deficit in primary progressive aphasia. Together, these data suggest that changes in tau occur followed by changes in atrophy, followed by changes in cognition. I want to transition now and provide an update on treatment and care. Let's start with some historical context. Prior to 2011, funding for Alzheimer's was relatively abysmal in comparison to other major diseases. In 2011, there was a major shift. The National Alzheimer's Project Act was signed into law by then President Obama. This law resulted in the National Plan to Address Alzheimer's Disease, which has 5 goals that are listed here. The most bold of which is to prevent and effectively treat Alzheimer's disease and related dementias by the year 2025, which is quickly approaching. The national plan has served as a catalyst, increasing research more than 7-fold and allowing scientists to work at a more rapid pace to advance basic disease knowledge, explore ways to reduce risk, uncover new biomarkers that I mentioned earlier, which enable earlier diagnosis and help with drug targeting, and to develop new potential treatments. One way that progress is tracked for this initiative is through Nih Research Summits, which are key strategic planning meetings tied to the implementation of goals of the national plan. The summits bring together a global and multi-stakeholder community, including government, industry, academia, private foundations, patient advocacy groups, and the public to further integrate translational Alzheimer's research. The most recent NIH Summit was held last month and can be viewed online for those who are interested. So what has the national plan done for treatment? Well, in 2012, the title of a scientific paper for Alzheimer's treatments read, Alzheimer's Disease Drug Development Pipeline, Few Candidates and Frequent Failures. This paper reported that there were only 5 unique therapies in clinical trials for AD. I'm happy to report that the landscape is much different now. In 2020, the number of unique therapies skyrocketed to 121 agents in clinical trials for the treatment of AD. And they're categorized in this complex figure here on the right, which is probably difficult to read. So let me give you a few of the stats. 97 of these 121 drugs are disease-modifying agents intended to change the underlying biology of AD. Over the past 5 years, there's also been greater diversification of the targets for drugs in the pipeline. And those are highlighted here by these colorful boxes over here that are then reflected in this graph over on the right. And greater integration of biomarkers into the development program, as well as there's also been advances in trial methodology. Trials are available for individuals across a wider spectrum, from those who are healthy, thinking about prevention of Alzheimer's disease, to those with severe dementia. This is important because we now know that changes in the brain can emerge years before clinical symptoms. Our clinical trials unit, led by Dr. Ian Grant, who will be on the panel later today, continues to identify and offer a subset of these trials for those who might be interested in participating. You might be wondering, what else can I do to avoid dementia? Although age, genetics, and family history cannot be changed, promising research suggests that other risk factors may be able to be modified to reduce the risk of cognitive decline and dementia. In fact, the 2020 recommendation of the Lancet Commission on Dementia Prevention, Intervention, and Care suggests that addressing modifiable risk factors might prevent or delay up to 40% of dementia cases. I rearranged this figure so it'd be a little bit easier to see the identified potentially modifiable risk factors organized into 3 categories: early life, midlife, and late life. Examples of modifiable risk factors include avoiding physical inactivity, avoiding smoking, higher educational attainment, staying socially and mentally active, and maintaining good blood pressure and a healthy diet. Individuals who take measures to reduce risk may still develop dementia, but they may develop it later in life than they would if they had not taken these steps to reduce their risk. Collectively, these data suggest that there are ways to potentially modify risks across the whole lifespan. Pharmacological trials, as well as behavioral and multi-component interventions, are underway to address these potentially modifiable factors. We have several programs at the center focused on optimizing quality of life, many of which are led by Dr. Darby Morehart, and some of which you heard about earlier today. Here I'm highlighting the Communication Bridge clinical trial from my lab, which uses technology to improve access to speech-language therapy care by connecting individuals with a diagnosis of PPA and their communication partners to a speech-language pathologist using video chat-based intervention sessions. It is supported by a custom web application that provides home exercises, video strategies, and a calendar feature for keeping track of sessions. Our approach has bridged geographic barriers and allowed us to enroll participants across the US, Canada, Europe, New Zealand, and the US Virgin Islands. The goal of this study is to help identify the most effective strategies to optimize quality of life. A pilot study of over 50 participants demonstrated initial feasibility and showed that it's possible to improve communication confidence and maintain those gains. We're currently running a randomized control trial, and we're on track to complete enrollment later this year and look forward to sharing the results afterwards. New directions and synergies. One of the key recommendations emerging from the National Plan to address Alzheimer's disease is to bring new perspectives and expertise to the complex challenges of Alzheimer's disease and related disorders research. Our center has been multidisciplinary from the beginning. In fact, sometimes we say we cover everything from cells to social work with our interest and expertise. But we continue to look for new partnerships. And I wanted to highlight the work of Dr. Kelly Jarvis. During her graduate training, she gained expertise in developing advanced cardiac imaging techniques. Recognizing that potentially modifiable risk factors for dementia include cardiovascular risk factors, Dr. Jarvis is now developing specialized imaging methods that will allow for imaging of the heart and the brain in a single session, something that has not been done previously, in order to study heart-brain linkages in Alzheimer's. And this is one of the first sets of pilot data showing that this can all be done in a single session. We're excited to hear more about her work in the future. Now we're going to switch gears entirely and to talk about heterogeneity and aging. So far, we've been focused on the pathologic trajectory of aging. But there is also a normal trajectory of aging, which is associated with some degree of cognitive decline. More than 10 years ago, we wondered if we could also identify and study individuals with little or no age-related cognitive decline. We call these individuals superagers and define them as individuals over age 80 with memory performance at least as good as individuals in their 50s and 60s. You may wonder, why would we study such individuals? Well, as a medical community, we've gotten good at increasing our lifespan with advances in healthcare, but our health span hasn't necessarily kept pace. I think superagers represent a good example of health span matching lifespan, and we want to know how are they able to do this. We also think that studying superagers can help us inform Alzheimer's research. In fact, this was another recommendation as a part of the National Alzheimer's Plan, was to understand all aspects of healthy brain aging and cognitive resilience to inform strategies for Alzheimer's disease prevention. Superagers also importantly help us to redefine expectations of what is possible in aging. So not just focusing on what can go wrong, but actually what people can still do and go right. And this is one of our superagers here, who at age 100 was interviewed by the local paper. And in that interview, it might be hard to see, this picture shows her beading. She's putting together necklaces while she's chatting during the interview. We were excited to help, be a part of her 106th birthday this past summer. And these are the cards behind her, more than 200 of them from her friends and family to celebrate this great achievement. And we look forward to celebrating her 107th birthday later this year. Studying superagers also helps us think about how to reduce stigma. This is another one of our superagers who loves to travel. And in the background here, I'm not sure if you can make it out, but those are actually penguins that she's on one of her great adventures seeing. So what are we doing to study superagers? We're taking a multidisciplinary approach to identify relevant factors to maintaining superior memory performance in old age. We have 4 biologic cores that I like to think about. So cognition, neuroanatomy measured by brain imaging, histopathology, looking under the microscope, and genetic factors. We complement those by looking at lifestyle, education, family history, and psychosocial factors. So what have we found so far? I'm going to walk you through a very quick overview of some of our key findings we've made. Superagers are able to maintain their superior memory performance over time. They don't just have a lucky memory day when they visit us. We see that superagers have less cortical brain shrinkage over time. On the top here is where there's atrophy in normal aging. And we see that compared to 50-year-olds. And down here, we see that superagers don't show that same atrophy. In fact, they have a region of the brain in the anterior cingulate, shown here in blue, that's thicker in the superagers than it is even in the 50 to 60-year-olds. When we look under the microscope, we see that superagers have 4 times more von Economo neurons than their peers. Von Economo neurons are large neurons that have been described in higher order species and have been linked to social emotional behavior. They've only been described in the human brain in the anterior cingulate and the frontal insular cortex. This work was completed by Dr. Tamar Geffen when she was a graduate student and continues in New Directions today under the leadership of Drs. Giola and Geffen. Genetics. We asked the question, do superagers just represent individuals who have unusually low AD risk? And the answer is no. We used a specialized genetic risk metric called a polygenic hazard score. We found that the risk for AD in superagers was similar to that of their cognitively average peers. So superagers' memory performance cannot be explained by having unusually low risk for AD. When we look at lifestyle factors, we see that more than 80% of superagers still exercise and lead active lifestyles. Their education levels range from high school to advanced degrees. Some have longevity in their families and others have a history of dementia. They also report strong social relationships, one of the aforementioned potentially modifiable risk factors. Our studies of superagers continue, and we remain excited about what superagers can teach us about expanding our health span. If you think you know a superager, please have them reach out and join our research. So I've taken you through some of the high points of aging and dementia research, both locally at the center and globally. I hope you'll agree with me that this is a time of great excitement and progress. I want to close my time today with the following themes in mind: resilience, creativity, strength, and gratitude. Our participants and our families continue to inspire us with their resilience and their creativity, their strength, as well as their ability to find humor and joy. This quote of unknown origin was in the signature line of a Care Partners email that I received, and I thought it embodied an interesting perspective. People who wonder whether the glass is half full or half empty miss the point. The glass is refillable. The images here on the right show artwork created by individuals living with a diagnosis of PPA that they created after their diagnosis. They highlight the creativity and resilience, but also maybe the refilling of glasses. I'm grateful that you chose to spend your time with our center today. If you are longtime friends of the center, we hope you will continue to stay connected with our community. And if you are new, we hope you will come back to learn more and become more involved in this community in a way that resonates with you.